Now we want to take a look at the structure of the plasma membrane. The plasma membrane is described by what we call a fluid mosaic model. It is a mosaic of components. It has a lot of different types of molecules in it, including phospholipids, cholesterol, proteins, carbohydrates. It is also fluid in nature. So these molecules can move around within the membrane. The membrane itself sort of floats, if you will, sort of like a wave on a surface. These molecules here can move across, they can move through, and so there's quite a bit of fluidity to that membrane. Let's take a look at the basic background of the membrane, which is composed of phospholipid. Phospholipids are what we refer to as amphipathic. That means that they have part of the molecule that loves water and part of the molecule that does not love water. So we have a hydrophilic portion, which is this head, and then we have a hydrophobic portion, which are these tails. Recall from our chemistry section that a phospholipid is made up of a glycerol molecule, and that's what we see right here. This is that 3-carbon chain. Attached to it, we have 2 fatty acid chains coming off here. Those are the tails. Those are the nonpolar regions. Because they're nonpolar, they are hydrophobic. Then we also have up here a phosphate-containing group. That is polar. So because it's polar, that is the hydrophilic portion that likes water. And then we can have other things recall attached to the phosphate group, and so that's symbolized here with an R. Recall also that this fatty acid composition can vary. And so sometimes we might have more saturated fatty acids and we have no kinks in those chains. And then other times we might have unsaturated fatty acids that allow that molecule to kink. Remember, when the molecule is kinked like that, it also makes it a little bit less rigid and a little bit more fluid in nature. The term saturated means that it is saturated with hydrogens. So you have carbons that have as much hydrogen on them as they can possibly take. That means that there's all single carbon-to-carbon bonds along here. If it's unsaturated, we have a double bond in here somewhere, which means that we've had to remove a few hydrogens in order to get that to occur. Phospholipids tend to form these bilayers. Even when it's not a live membrane, phospholipids will form bilayers. This is showing a single phospholipid sphere, which we see over here as this micelle. But typically what will happen is you'll end up with a double layer. You have the phosphate heads here on the inside, and then you have the phosphate heads here on the outside, and then these bilayers point towards one another. You also can form this bilayer sheet that we see down here. So they can arrange in a sphere or a sheet, and typically will form these bilayers naturally. When they form these bilayers, then the polar heads will face outward and the fatty acid tails will face inward. This is going to be in an aqueous solution. So imagine this is completely surrounded by water. So these phosphate heads are facing the water on both sides, and now these fatty acid tails are away from the water. They're next to each other. They're all nonpolar. They're near other nonpolar molecules, so that makes them happy. And then these polar heads are facing the water, which is also polar. Embedded in this phospholipid bilayer, we have our proteins. We have a variety of different types of proteins. We have integral proteins. These integral proteins are completely embedded into the bilayer. They're not just sitting on the surface. Now, they don't have to go all the way through. They can go all the way through, and then if they do go all the way through, we call them transmembrane because they cross the membrane. But they also can be non-transmembrane, but still embedded in that bilayer. So these red ones here are all considered integral proteins. Some are transmembrane like these, and then this one is a non-transmembrane, not going all the way through. We also have peripheral proteins, which are only on the surface. So you see these sitting sort of on this outside surface. So those are peripheral proteins. They might be on the bilayer, or they might be actually attached to another protein like we see here, but they're only on the surfaces. These peripheral proteins can be on both the inner or the outer membrane surfaces. They can function as enzymes or to anchor objects, or they can also be transport carriers. And we'll see functions a little bit later. The integral proteins that we see here going into the membrane and often spanning across the membrane have a variety of different functions. They typically then, because they go into the membrane, will have one or more regions that are hydrophobic because they are here in this fatty acid tail area, so they're hydrophobic in there. And then they'll have other areas on them that are hydrophilic. And you can see a variety of different functions we have here, which again we'll talk about later, but they can act as receptors, they can act for recognition, enzymes, transporters. The location of the protein and then the number of different regions they have will determine how they arrange themselves in this bilayer. We can see a variety of different arrangements here. You don't need to know these details of these arrangements, but they do have a variety of different arrangements that then correlate with their function. Next we have carbohydrates. Carbohydrates are the third major component of the membrane. Most of these are oligosaccharides. So we talked before about monosaccharides and polysaccharides. Mono was a simple sugar of one sugar unit. Polysaccharide was many. Oligo means it's a short chain. So it's not a super long polysaccharide like cellulose. It's a much shorter chain. So what we see here with these oligosaccharides, you can see these are showing individual glucose units attached to a protein. And then here we see some sugar units attached to some lipids. So we can have glycoproteins, or we can call them proteoglycans. Usually if you call it a glycoprotein versus a proteoglycan, it really depends on which part of the molecule is bigger and which part is the modifier. So if it's a proteoglycan, it's usually a longer carbohydrate and a smaller protein. If it's a glycoprotein, it's a bigger protein and a shorter carbohydrate. So they can be attached to the lipids or the proteins there. These are typically located on the exterior surface of the plasma membrane. Before we saw where we had molecules on both sides of the membrane, the inner side, which if this is the cytoplasm down here, this is the inner surface of the cell here. This is the outer surface of the cell up here, and this is the extracellular fluid that is surrounding the cell. These carbohydrates are on that outer surface of the cell, not the inner surface. These typically will function in cell-to-cell recognition. I'm the same type of cell, I belong to this body, that kind of recognition. And then also for attachment. If you think about it, these are carbohydrates, they're somewhat sticky, and so it can cause cells to stick to one another. Next we have glycoproteins. So these are the proteins that are modified by having this carbohydrate there. We can see a variety of different glycoproteins here. So it allows cells to recognize one another, signal to one another, stick to one another. And when we talk about protection, we're looking a lot about that immune response that we see cells exhibiting. So for instance, our immune system has T cells. T cells have CD4 receptors on them. This is a particular type of glycoprotein found on T cells. HIV enters T cells because it recognizes that CD4 on our T cells. So that allows HIV to enter those cells. That is one way that certain, say, viruses affect certain types of cells, is because they recognize very specific receptors on those types of cells. So while these recognition molecules allow our T cells to identify cells that belong in the body versus cells that do not, and to attack cells that do not belong in the body, it also offers an opportunity for invading cells to overcome some of those responses by being able to find those cells and get inside those cells. This outer surface that contains a lot of these carbohydrates is called the glycocalyx. We can see that here, and it has a lot of different carbohydrates attached to proteins and lipids. Now let's take a look at membrane fluidity. The membrane needs to be flexible, so it needs to be somewhat fluid, but it also needs to be somewhat solid. If it's too fluid, it can't maintain the structure of that cell and the integrity of the cell, but it still needs to be fluid to allow materials to pass through it and to have some flexibility. So the fluidity is affected by three different things primarily: the phospholipid type. Recall that phospholipids have those fatty acid tails, and those fatty acid tails can be either saturated or unsaturated, and the degree of saturation can vary. And remember, the saturation allows the straight chains to pack more closely together, and an unsaturated fatty acid with the kinks, they don't pack as closely together. So a membrane that has more saturated fatty acids as opposed to unsaturated would be a little bit more solid, a little bit more inflexible and rigid. One that has more unsaturated fatty acids would tend to be a little bit more fluid and a little bit more flexible. Temperature also will affect membrane fluidity. Cold temperatures tend to compress molecules, making the membranes more rigid, and then warm temperatures tend to make molecules spread further apart, making them more fluid. Now, one of the things you find often is that there's a correlation between phospholipid type and temperature. Plants, for instance, that live in cold environments typically have a little bit more unsaturated fatty acids in their membranes because that allows them to maintain a little bit more fluidity in cold temperatures. If they had only saturated fatty acids, those cold temperatures, their membranes would be a little bit too rigid. And it's one of the reasons why we often recommend oils from plants that are more from colder regions as opposed to warmer tropical oils, because the amount of saturation in the plant cell membranes is less in a cold climate plant as opposed to a warm climate plant. Now, there's other aspects as far as essential fatty acids that doesn't consider, but just with respect to the saturation, we do tend to find a higher degree of saturation in those tropical oils. Cholesterol is another component of membranes that affects the fluidity. What cholesterol does is it's embedded in that bilayer in between some of those phospholipids, and it kind of buffers the fluidity. It can allow it to be a little bit more fluid or a little bit less fluid, but depending on the temperature. So if it's cold, it can keep it a little bit more fluid when it would tend to get a little bit more rigid. And then if it's hot, it can make it a little bit more stable and rigid as opposed to becoming too fluid. So it's analogous to the way a buffer works in regulating pH, only here it's a molecule that's regulating fluidity. Here we'll watch a brief video on this membrane fluidity.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Let's take a look at the concept of membrane asymmetry. The two sides of the membrane are not symmetrical. This is the cell membrane. The inside of the cell is down here at the bottom, and then the outside of the cell is up here at the top. So this is the extracellular fluid that it's bathed in. This is the cytoplasm down here. This asymmetry means that the side that is up against the cytoplasm is going to look and function a bit differently from the side that is up against the extracellular fluid. So for example, the interior proteins that we see here on the membrane will typically be connected to some of the cytoskeleton that helps to anchor the membrane a bit to the cytoskeleton. Some of these exterior proteins will bind to the extracellular matrix. So there will be proteins out in that extracellular matrix, especially in tissues like cartilage and tendons and ligaments, where there's a lot of extracellular matrix that has a lot of protein in it. These proteins will often form fibers, and so the cells can attach themselves to those fibers. The glycoproteins that we see here and the glycolipids, remember, they can also bind to substances that need to be imported in the cell, and they can also bind to other molecules as receptors and cell identification. So there's a lot of things that occur outside the cell more for identification, adhesion to other cells that are happening on this outer surface as opposed to this inner surface. So there's definitely the two sides of the membrane look very different from one another. So that gives us a brief overview of the structure of the plasma membrane. Next, we'll take a look at the function of the plasma membrane.